So welcome back. So now we'll have the second talk of today. So this is going to be the second lecture by Professor Rahul Siddharthan, who's already gave one lecture before. So this one's going to be on string alignment, evolution, phylogenetics, and how languages evolve. As to like how you can use insights obtained from computational biology to linguistics. So without further ado, let me invite Rahul to give this talk.
Mic check.
Is that okay? Yeah. Okay, so my original plan was to talk about string algorithms because that seemed to be relevant to finding common substrings in texts and things like that, which epigraphists do. But I won't talk about that very much, but instead I'll talk about essentially, let's start with how DNA evolves. So DNA, and this is perhaps a model for how texts in general might evolve, but for DNA, as you know, your genome consists of 23 pairs of chromosomes. In the case of males, it's 22 pairs and an X and a Y. And these get replicated during cell division. They also get replicated during meiosis, which is gamete formation, but there is a sort of twist there where there is something called recombination that also happens, where the chromosome that goes into your gametes is not an exact copy of any one chromosome you have, but a sort of combination of the two. So as DNA evolves, as it replicates, errors can occur. It can change in some ways. So the most common is single nucleotide substitution. So if you compare any two genomes of any two people here, they'll differ by about 1 in 1,000 nucleotides, partly because of replication errors, partly because of mutations due to cosmic rays or other mutagens. These things happen all the time. You can have insertions, deletions, meaning you might have a gap or you might have some extra DNA in a region of your chromosome where the standard genome or somebody else's chromosome doesn't have. You can have segmental duplications, and at a sort of, this is very common in cancers, but also at a larger evolutionary time scale, this is believed to drive evolution. You can have duplication of entire chromosomes, even of entire genomes. The yeast genome is believed to have had a whole genome duplication long back. You can have recombination, which is when different pieces of DNA combine. So what I mentioned, crossover in meiosis is one example, and so on. And we won't be talking about any of these, but when aligning two sequences, typically two DNA sequences or two protein sequences, which are fairly short sequences, we look mainly at these two processes. And the idea is that DNA doesn't arise by itself, it evolves from some parent. And all DNA is believed to have evolved from some common ancestor. So now given two sequences, there's this term in biology called homology, which means that if they're homologous, then they derived from a common ancestor. So basically the idea is, given two sequences, can you, right, so for phylogenetics, this is what we do. And this is a typical tree of life that you might have seen from, this is from more than 20 years back, but there's already it's not terribly readable, but core data is basically where we are. It is things that have a spinal cord. The various kinds of worms and whatnot here. The fungi are here. The plants are here. Bacteria are here. Archaea are here. So from the tree of life, you can tell that bacteria form a group. So this sort of branching structure, which we already saw in Nisha's talk and you're probably familiar with, is kind of indicative of how things cluster together. And we'll talk a little bit about how to make these diagrams. But you can see that chordata form this cluster here, and bacteria form this cluster here, and plants form a cluster here, and so on. So how do you build a tree like this from DNA, let's say? So the basic, so I'll start with sequence alignment and then with how to do this. So the sequence alignment task is given two sequences, match the bases that are likely to be derived from a common ancestor. So for example, these might be two sequences you have. Somebody gave you two sequences like this, but they asked you to align them and your answer is this. So what this means is that everything that's vertically aligned, you believe is conserved from some common ancestor. And whatever is not vertical aligned, like a dash basically means you've padded out one sequence to satisfy the alignments. The dashes don't actually exist in the sequence, but it's either that these 4 nucleotides got inserted here or they got deleted here. With just a pairwise alignment, you can't tell which is which, so you call it an indel, insertion or deletion. If you have a multiple alignment, you might have a case where 9 out of 10 have something here and 1 doesn't, then it's definitely a deletion, or it might be the other way around and it's an insertion. So let's talk a little bit about how this is done. Given two sequences, how do you find an alignment? And computationally, this is actually the oldest problem in bioinformatics. It was in a paper by Needleman and Wunsch in 1970. The idea is find the alignment that optimizes some kind of scoring function. And basically, the way you do it is that you think of it probabilistically. If you have you have an A here and an A here, and they both came from a common ancestor, what's the likelihood of seeing two A's jointly? And that is given by a joint probability. Small b's I'm using for bases in this case, and or nucleotides. So what's the probability of jointly seeing them? It is this. What's the probability of independently seeing them? It is just the product of the probabilities. So the ratio is the likelihood ratio, and the it's the probability ratio, and or the odds, and the log of it is the log odds. And this is a reasonable score to use, and this can be done additively across all pairs of nucleotides to give you a total score. And that's a reasonable thing to optimize. But of course, that's not all there is. You also have these gaps, and how do you deal with them? Basically, you kind of want to penalize them, because every gap is something you don't want. It sort of disrupts the sequence. So the standard ways you do it are with a linear penalty where you have some constant d, and for a gap of length L, the penalty is just L times d. So it is 3 times d here, 4 times d here, and just d here. You could also have an affine penalty where E is a bit smaller than d, and the logic is that it's expensive to introduce a gap, but once a gap exists, it is not that expensive to extend it further. So this is the Needleman-Wunsch algorithm that does this. So the space of all possible alignments, every possible way you could match two bases or insert a gap and so on, is kind of exponentially large, but it turns out you don't have to search that space. You can build it up slowly. It's an example of what computer scientists call dynamic programming. And the idea is that two sequences x and y of length L1 and L2, you make a matrix. I'll illustrate it in the next slide. This matrix gives the best alignment for the first I letters of x and the first j letters of y. So, and then it turns out you can calculate each element of this matrix from the previous elements of the matrix. So let's just so I'll just go to this. The idea is that each element of this matrix contains the best score. So supposing you're aligning these two sequences, this element would be how do you align the single letter A with the three letters ACT, what is the best possible score you can get. And here we are using a scoring matrix where a match has a +1, a mismatch has a -1, and the gap has a 0.5, minus 0.5 really. And so the idea is that if you wanted to know what this element is, for example, you could get this element by getting the best alignment of, so this element corresponds to the best element of ACTC and ACC. And this could be done by getting the best alignment of AC and ACT, which was here, and matching these two. And since C and C match, you get a 2.5 because this was a 1.5. You could also have got it by taking the best alignment of AC and ACTC and inserting a gap in the second sequence. So this would be 1 minus half, so it would be half. Similarly, you could go get it from here and insert a gap in the other sequence, and that would be again 1 minus half, which is minus half. You compare those three numbers, 2.5 is greater than half. So you put in the largest number you get and an arrow to the cell you got it from. And you fill this entire matrix row-wise in this way until you reach the bottom right. And once you reach the bottom right, this is the score of the best possible alignment of these two sequences. And if you follow the arrows, you will get the alignment. I think I did not show the alignment you actually get, but if you follow the arrows, you can tell that the last 4 letters are actually aligned exactly. And at this point, you are taking, you're basically making an insertion, or you're making an insertion in the first sequence. So there's a T here and a dash here, and then you have an. So this is the output of your program, and you can do this for much larger sequences. Obviously, the complexity of this is L1 times L2, the product of the lengths of these, both in space and in time. So this is a sort of bread and butter algorithm for pairwise sequence alignment. So I won't go much further into that, except to say that there are highly optimal ways to do it, and there is a variation of this if you want a local alignment. Supposing you have 2 very long sequences and you just want to find something that is similar somewhere in the middle of those sequences, not wanting to align the entire sequence. So for example, you have a long sequence and another long sequence, and you care about aligning whatever is conserved. This is the conserved region, and you don't want to penalize whatever is hanging on both sides. Then you can, there's a simple modification of this algorithm called the Smith-Waterman algorithm. You make the same F matrix, but now you don't let any element become negative. If it is negative, you set it to 0. For this particular case, there were a few negative elements. So any negative element, you set them to 0, and without any pointers. And when you do the backtrace, you don't start from the bottom right, but from the largest element you have found in the matrix. So that would probably be the, so the effect of this basically is that up until you reach about here, your score is bad, and therefore you keep resetting it to 0, but there is some cell here where you start making the alignment and it's good. And you keep doing that until you hit a 0 again, or until you hit the maximum value. And after that, it's decreasing, so you're penalizing the alignment you've already got. You start from where you've got the best score and go backwards until you hit a 0. That's your best local alignment. So potentially, maybe that's a algorithm that could be used in finding common substrings in other contexts. So it finds inexact matches because you can have mismatches, you can have gaps and so on, but it is still the best local match. So for proteins and things like that, it's very important, and probably it's usable in other contexts also. So with that, and yeah, please stop me if you have any questions at any time. I just wanted to introduce this before going on to phylogenetics. So as I said, we are not talking about recombination and so on, but even in terms of the two processes we saw, we are now going to ignore deletions, and we're going to look at only gapless alignments. This is completely fictitious example, I just made it up. But how would you get a thing like this? What you would do is you would get an alignment, multiple sequence alignment of these 5 species, not of the whole genome. You would probably do gene by gene. You'd focus on some typical genes that they look at. And then from those alignments, you basically crop out all columns where you have indels. So for example, if this was your alignment, you just block out this part completely, the entire column, and you concatenate everything else together. So you join this to get a gapless aligned block. So this now tells you, it gives you a list of homologous nucleotides in each species. So what kind of information does this give you? The basic thing it could give you is the number of mismatches between 2 species, which is kind of a proxy for the distance between them. So if you look at human and chimp, in this case, there are 2 mismatches between them. If you look at mouse and rat, again, there are 2 mismatches. But if you look at, let's say, chimp and mouse, you have 1, 2, 3, you have 3 mismatches in this case. If you look at cow, you have many more mismatches. So if you were to plot a tree based on these, you can imagine that this is what it might look like, that human and chimp are the closest, mouse and rat are also close to each other, and then these 2 are kind of separated, and cow is an outlier, it's somewhere else. So how do you formalize this? We'll so what is the phylogenetic distance? So the crude distance measure is what I just said, it's the number of mismatches between them. But it's better to use these mismatches to estimate the evolutionary time, the amount of time that has that these species have diverged. And you could assume a constant mutation rate. We'll come to that. So the evolutionary time is monotonic in the number of mismatches roughly, but not linear, because the number of mismatches saturates at about 75%, because 25% will always match by chance. I'm talking about DNA. So since there are 4 nucleotides, you'll get 1/4 of them matching just by chance. So just to say in advance, a lot of what I'm saying seems to be carried over to the linguistics field, except that you don't have 4 nucleotides, you have many different kind of word units and so on, but they do talk about mutation rates and evolutionary times and things like that, and as I will be talking about later, they are actually using, some of them actually use phylogenetics program like BEAST, which is written for DNA but written kind of flexibly. They just modify it to run it on languages instead. And so all this stuff has analogs in that field, but I'm not an expert in that field, so I can't tell you exactly what they're doing. Okay, so the idea is that you had an ancestral sequence which had some collection of nucleotides. Maybe it could be just the consensus of these, since they are fairly diverse, or it could be something else at each position. And it has been evolving. This is the common ancestor of these species, and it has been evolving over time. So the simplest example is what's called the molecular clock hypothesis, which is that everything basically is evolving at the same rate. So so there's a constant mutation rate, and the number of mutations you expect along any branch would be roughly the same. So from the common ancestor. But of course, these 2 are closer because they've been diverging for less long and so on. So how do you estimate the evolutionary time, the sort of geological time, which would be this axis, from the number of mutations? So for that, you need an evolutionary model. And here is, and then how do you evaluate the quality of your tree? So I'll come to likelihoods later, but essentially the way it is done is you can have an instantaneous rate matrix, which is a rate of mutation from nucleotide A to nucleotide B. So it's written, you can read it as R of beta given alpha. And from this you can calculate a transition matrix, which is the probability of seeing beta at a later time given alpha at a previous time. So that's, have I actually done that? I think I haven't described it in detail, but okay, so let's, so the picture is kind of like this. You have 4 nucleotides, ACGT. So think of each having a population in sub alpha, where alpha could be A, C, G, or T. And then you have a rate equation, how does this population change with time? And that is the rate of mutations from other nucleotides into this nucleotide. So it would be the rate of, sorry, where beta is not equal to alpha, minus again beta is not equal to alpha. So the rate at which other nucleotides which are not alpha turn into alpha minus the rate at which the nucleotides that are alpha turn into something else. And these look like matrix sums except for this not equal to thing, but you can get rid of that by defining R alpha alpha, which is the diagonal element of the rate matrix, is defined as minus sum over beta not equal to alpha or beta alpha, which is basically this guy. And with that definition, you can write this as dN by dt is equal to R times N, where R is the rate matrix. I'll write N as a vector and R as a matrix. And the solution for this is N of T so the exponential of this matrix R alpha beta is the transition matrix T beta alpha of time. So that's the basic principle here. And then the rest of the game is basically choosing the rate matrix. But before getting into this, we'll talk about some simpler ways of doing phylogenetic trees, which I think have also been done in linguistics. So these don't require this kind of evolutionary model, but just a distance matrix. So in fact, this is basically the same as what you call agglomerative clustering in clustering examples. So given a bunch of points in some abstract space, and in this case, let's say it's actually the Euclidean distance in this two-dimensional plane, what you do is you identify the points. This figure is from Durbin's textbook, I should have acknowledged. You first put every point in its own cluster. So the first step is really 1, 2, 3, 4, 5. Each point is in a cluster by itself. And the first step is you identify a pair of points that are the closest, pair of clusters that are the closest. Initially, each point is just a cluster, and you identify that this is the closest. So what you do is you combine 1 and 2 into a cluster and give that a new label, which in this case is called 6. And now you have 4 points in your space. This is now 6. And so you have points 3, 4, 5, and 6. And now you identify the closest points that are remaining there. And that ends up being 4 and 5. So that gives you a thing that looks like this. And you call that 7. And then, so ignore the circle for now. What you had is this. And now the closest points here are between this cluster of 2 points and this one. And that gave you this one. And finally, you join the whole thing together, and that's what you get. And the way you calculate these distances is by, so the distance between any two clusters is the average of pairwise distance between all pairs of points among those clusters. So if it's just 2 points, it's just a distance. If it was, for example, this cluster and this cluster, it is the average of the distances 1, 4, 1, 5, 2, 4, and 2, 5. So that is the distance between cluster 6 and cluster 7 in this language. So that works pretty well for basic tasks, and if you just want to get a general picture, it's probably the first thing you'll do. But it can also fail, and it makes this key assumption of this molecular clock, which is that everything is evolving at basically the same rate. So there are a bunch of other things that people do, which is called, one is neighbor joining, so the other assumption here is additivity, that the distance between 2 points, between 4 and 7 and 5 and 7 adds to the distance between 4 and 5. So the branches are additive. If you assume that, but drop the molecular clock hypothesis, you can have, there is a method called neighbor joining that works. And in this case, you get a rooted tree because after you've joined everything, the root is this node where you joined everything together. But in principle, in phylogenetics, you can have an unrooted tree, meaning you don't know where the root is. And you can establish the root by introducing an outgroup, which is a different species from somewhere else. So neighbor joining basically does that. There's a method called parsimony, which I won't talk about, but essentially it is given the sequences at these leaves, can you infer the sequences at the nodes that result, well, so you're trying to guess both the topology of the tree and maybe the sequence of the nodes, but what you're trying to minimize is the number of mutations that you got from the ancestor to the leaves. And it turns out there are reasonably efficient algorithms to do that. So I won't talk about that either, but the most common methods and the ones that I think that have been applied to linguistics are maximum likelihood and especially Bayesian methods, which are sort of extension of maximum likelihood. So let's go back to the transition matrix. Yeah, so basically this is what I wrote on the whiteboard. Sorry, I had a last minute change of topic, so I'm not very organized with this talk. But essentially, the transition matrix is the exponential of the rate matrix, which is what I wanted to say. And now the question is, how do you choose a rate matrix? Let the stationary distribution at any locus be pi vector, which is, so this is the fraction of nucleotides, or the fraction pi c pi g pi t, which could be 1/4 each. Then for a time reversible matrix like this, this condition is required, which is called detail balance. Essentially what this is saying is that the stationary distribution doesn't change under the application of t. You also have a property of t, which is multiplicativity, which means that essentially what this means is that if you have a point time, so you had an ancestor beta, and it evolved over time t2 to gamma, and then it evolved over time t1 to alpha. Then the transition matrix from beta to alpha over time t2 plus t1. No, I did it slightly wrong, sorry. This is gamma, this is beta, and this is alpha. So basically, the transition matrix from here to here is the product of the matrices corresponding to this and corresponding to this. So you should read this product backwards, really, then it's a matrix product. And there's something called a pulley principle, that the root of a rooted tree is not uniquely determined by the tree alone. So essentially, when you had a tree like what I showed you earlier, I drew it like this, and I picked the root here. But if you talk about the transition probabilities, you could equally think of, so you have a transition matrix from this point to this point, and also from this point to this point. So let's say this was alpha, this was beta, this was gamma. You could equally think of beta mutating in this direction to alpha to gamma. You don't know which way it went from the math alone. So essentially you can reroute the tree so that this is the root, or the root is somewhere here, and so on. It's like a pulley, you can rebalance the tree. So that's kind of important. Yeah. Yeah.
Considering n alpha, n beta, and alpha, they belong to ATGC, right? So that does that mean that you're not talking about, you're just disregarding the sequence, as in what comes
sorry.
after
So
the other?
this was meant to be a population. Let's go back here. Yeah.
Yeah.
So think of a population where the fraction of nucleotides is n alpha. You're not talking about an individual, but you're talking about probabilities. The easiest way to think over that is in the frequentist way. So think of a population.
So it's not at a particular position, but
It is at a given locus. It's a particular position.
also you.
So then you have to multiply this over all positions. I'll come to that. So if you're talking about one particular position, and this T matrix is for that one particular position, you'll have similarly a T matrix for each other position, which could be the same across positions, or it could be different because different positions are under different selective pressures. And we're assuming position independence. So the final answer would be a product over all positions. There's no dependency among the positions. Okay, thanks for that. And that assumption will carry over into linguistics, I believe. Yeah, so how do you decide what to pick for R? So these are the most common choices. And again, from what I'm reading, some version of Jukes Cantor is used in linguistics. Basically, this is saying all mutations are equally likely. That means all off-diagonal elements of R are equal. So R looks like mu, 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 and so on. And the diagonal elements are minus 3 mu. And so on. So, and then because in DNA you know that transitions are more common than transversions, you could differentiate these two. Both Jukes Cantor and Kimura result in a uniform stationary pi, which is not important for our case. And there are more general models. There's something called the general time reversible model, which has a different rate for every possible mutation, but it's still symmetric. And then to get non-uniform stationary pi, you will need an asymmetric rate matrix. So I don't know what the analogy with that would be to linguistics, but let's ignore that. Okay, I'll skip these things. I thought I'd remove these slides. Okay, so the idea is that to evaluate a possible tree, so just to backtrack, you have a bunch of species, a bunch of sequences corresponding to each species, and these are hypothesized to have evolved from a common ancestor. And now you're trying to guess what the tree is that links these species. And there are two properties of a tree. It's topology, which is how things are linked together, which is what UPGMA gave you, for example. So this is one possible topology, but you could have a topology where A and T are neighbors and C is further away, and so on. So actually with 3 species, there's only one topology because of the pulley principle, but just ignore that. But the other thing is these branch lengths. And okay, so I think I skipped a slide somewhere, but if your mutation rate is mu, Q is e to the minus mu t. That's the definition here. So if the branch lengths are, so Q sub I, sorry. T sub I. So that's what these Qs here mean. It's sort of reverse evolution of the branch lengths. And it's like a conservation probability, essentially. The probability that beta is the same as alpha. Okay, so what you're asking is what is the likelihood of seeing these particular nucleotides that you do see given this tree topology and given these branch lengths. And that's the left-hand side. And the right-hand side is the answer. So I'll start from going backwards from this term. So if this was a beta, then the probability of seeing A is T A beta given this branch length Q1, and probability of seeing this C is T C beta given this branch length Q2. But what is the probability of seeing a beta here? That depends on what this alpha was. So it's T beta alpha with this branch length Q4. And you don't know what beta is, so you sum over beta. And then that's one piece here. And then what is this probability of seeing a T here? It is T alpha given this branch length Q3. And now you don't know what alpha is, so again you sum over alpha with the background probability pi alpha. So that's what the likelihood is for this tree for this particular set of nucleotides. So again, going to Anurag's question, this is for one locus, and the likelihood for the set of sequences you have will be a product of this over all loci, like a product over all positions in the aligned sequence from 1 to L, whatever it is. So this is the formula. How do you calculate it? Felsenstein in 1981, I think, gave an elegant recursive algorithm that maybe I shouldn't go through, but okay, I think I'll skip this part. But essentially, the idea is that you can calculate a partial likelihood for each. So the likelihood for the full tree, you can read it from here. It's the likelihood of this piece, is what I wrote here, times the likelihood of, so this is basically the likelihood of this piece given beta. And then you have to, so you basically in the algorithm, you define this thing, the likelihood of leaves below nodes by, given that the nucleotide node I is alpha. And you initialize it for the leaf nodes, and for the non-leaf nodes, you calculate it recursively using this formula. And so that can be implemented very easily on a computer, and it's linear in the number of species and in the number of sites. So it's a pretty good algorithm. And it's kind of the bedrock of phylogenetics. So I just wanted to mention that. And then how do you actually infer a phylogenetic tree from sequence data? As I said, you need to infer the topology and you need to infer the branch lengths. And the approach is usually a Markov chain Monte Carlo approach, which is you sample the space of tree topologies. For any given tree topology, you can then calculate. So you have a tree, and you have a set of branch lengths here, Q1, Q2, Q3, whatever it is. But for any tree and any branch lengths, you know how to calculate the likelihood. But you want to maximize the likelihood. You want the maximum likelihood tree. So you calculate the branch lengths of this given tree using some maximum likelihood optimization algorithm. And then what you want to do is explore the space of trees. So you've got a tree, you've optimized the branch lengths. What you need to do is you need to change this tree into a different kind of tree using some kind of move, like for those of you familiar with simulations in physics, it could be like a spin flip. In this case, it's like taking one subtree like this and moving it somewhere else. So there are certain moves they can do that are guaranteed to let you explore the space of trees, and then you either, you do that and you re-optimize the branch lengths and recalculate the likelihood, and you accept or reject as per the Metropolis algorithm, which essentially is, if the likelihood is higher, you accept. If it is lower, you still accept with the probability based on the likelihood ratio. And that is basically guaranteed to explore the space of trees with a probability proportional to the likelihood. If you let it run long enough, and if your tree move set is ergodic, meaning you can actually reach all possible trees, then you can look through all possible trees. And so that's basically the way it is done. You can move to a Bayesian method to do it, which is basically, instead of the likelihood, which is this, you can look at the posterior probability, which essentially means you got a prior on the tree. And that means on the tree topology, because the branch lengths are, you'll still go for maximum likelihood for those. but you may think a priori some topologies are likely or not likely or impossible. So your prior can take care of those. So in the linguistics context, you may know for sure that certain languages are related. You don't want to sample trees where Tamil is grouped together with Greek or something. So you can rule out whole classes of trees using an appropriate prior. So yeah, so that's basically the idea here. These phylogenetics programs are very sophisticated programs that use Markov-Chain Monte Carlo sampling and likelihood calculation using Pulse-Einstein's algorithm and a clever move set to explore the space of trees and find the most probable trees. And there are a few things that I didn't mention that depending on the sort of sequences you select, you can actually get slightly different trees. And if you, so you might want to know what is sort of the support for a given branch, and sampling can help you do that also, in that while you're sampling, certain parts of the tree might be very fixed, but certain parts might flop around, and those kind of tell you that these are certain and these are less certain. Okay, so with that last few minutes on what little I understand of the linguistic analog to this. So typically the analog for nucleotides is words, but it's not like you have ACGT for words. You have an insane number of possibilities. And homologous nucleotides would be cognate words, as in the word for and or the word for one or whatever. You might have a list of words, and you can think of the list of words like a sequence of nucleotides, and the alignment is basically the columns carry the same meaning across languages and the rows are different languages. And how do you calculate likelihoods? So maybe there are people here who know something about this, but I don't. So, but they do seem to calculate likelihoods of trees in some way. And there's a bunch of papers for a long time. So this is kind of interesting. I was just looking around for what people have done, and this one is from Nature in 2003. This one is from Language in 2015. And you might know that there are two hypotheses for where the Indo-European languages originated. They originated in the steppes or in the Anatolian region in modern Turkey. And this paper supports this, and this paper supports this. And I'm not expert enough to tell who's right, but so we'll have some more comments on that. Similarly, people have looked at evolution of the Dravidian language family, extended it to cultural evolution. They argue that you can use language trees to test hypotheses not only about natural history and diversification, but about cultural adaptation and stuff. And actually, there is a question. Why do languages fit a tree model well? You would think that loan words are extremely common in languages, and in ordinary spoken Tamil, you use a lot of Sanskrit words, even though it's from a different linguistic family. So, of course, the linguists sort of take care of all that. But even in ancient times, we have reason to believe that there was some crosstalk between Dravidian languages and Sanskrit. Anyway, so this is a program that builds upon BEAST, which is a software tool for phylogenetics. They say BEAST is sort of specifically written to be extensible in many ways. So what they've basically done is written a tool to generate configuration files for BEAST and then run BEAST on that. And they use this Glottolog language catalog and so on, and they have a tutorial on how to use it. Yeah, so this is the tutorial. But essentially, you have to set up BEAST. You have to, and in the tutorial, they have a sample cognate judgment file, which is this file. I think it's here. So this is what the file looks like. It's a CSV file, comma separated value. I'm just showing it as a tabular format. So it has something like 100+ words and 20-ish languages. For each word, it has the IPA pronunciation in that language, and I haven't figured out what value means. But for example, if you look at the word all, many of them are cognate to all or to, but in Hindi it is sub, and in Romanian it is tot. So anyway, you can, they have 100 such languages, and in some cases you will find that the word for some word in Hindi is actually similar to the word in some European languages, but in most cases it's different. Whereas if you'll find that Dutch and German are almost always very similar. English is often similar to Dutch, but some German, but sometimes to French. Anyway, so if you run this tutorial, they say you will get some tree that looks something like this. So if you look at this, Armenian and Greek are kind of an outgroup, and Hindi is another outgroup. And among the others, these Slavic languages cluster together: Bulgarian, Czech, Polish, Russian. Then the Germanic languages, Dutch, German, English in one cluster, Danish, Norwegian, Icelandic, Swedish in another cluster. Then the Romance languages, which are French, Italian, Portuguese, Spanish, and Romanian in one cluster. So interestingly, from this limited repository, French looks like an outgroup compared to all of the others, which might be surprising to given the geographical location and so on. I think it's just because French pronunciations are so different. So if you just go by IPA, that's what happens. That's about all I have to say in this talk. So these are the caveats. So essentially, in biology, you know that phylogenetic trees are very sensitive, and if you look at published trees for example, yeast species or things like that, you'll find that papers published 5 years apart might have very different trees for some regions. And we might have similar concerns for linguistic trees. The choice of your corpus might be very important. In biology, we know that different parts of the genome evolve at different rates because of selection pressures. So some genes are highly conserved from yeast to humans, and other genes just vary a lot. So likely it's true for different words in different languages too. And there may be some cultural context, like in some cultures, certain words may be more important and more fixed. In other cultures, it may be a different set of words. And as I said, horizontal transfer is common. And so I'm guessing the linguists who do these things are first experts in linguistics, and so they know all these issues. But one should not use algorithms as a black box. So if you're using something like Beastling, I think it's good to know exactly how it works, as well as what kind of data you're giving it. Yeah, so I don't know whether this is like useful to anyone working on linguistics in this audience, but or whether anyone is actually doing these things already. But okay, thank you. Yeah. Nisha has a question, I think. Sorry?
About the string edit distance.
Yeah.
Can you just...
Okay, so what I showed was the Needleman-Wunsch algorithm. The edit distance is a kind of simpler thing. So edit operations are change a letter, insert a letter, and delete a letter. So in the edit distance problem, each of these has a score of +1. So if you just have change, then that's called the Hamming distance. That is, so for example, so the Hamming distance and also the edit distance between these two is 3, because it's this and this are different and the rest is the same. So the edit distance problem would be if you're also allowed to remove letters, insert letters, and so yeah, what I showed you was an example of that, but the scoring was different, that's all. So I'm calculating the edit distance between these two sequences. but instead of a scoring of 1, so for edit distance this would be 0, this would be 1, and this would be plus 1. Here this is plus 1, minus 1, this is actually minus 0.5. But the minimum edit distance will still be this, which is 1 insertion. And the algorithm is essentially the same. Yeah.
Thank you.
Thanks.
Any other questions or comments?
The Beasling example that you showed, it clustered on the basis of the IPA pronunciations, right?
I'm not totally sure, but I think so.
Okay.
But I don't know what those columns on the right side are, these
The
values.
value columns, okay.
So just had the idea of talking about this yesterday, and I messaged Sita to ask, is it okay? So I haven't gone too deeply into it myself.
All right, yeah, that's...
Yeah.
There's a question from...
Who in India is working in this area? Are you aware of anybody?
I have no idea, but maybe Bhaskar back there or Mayank knows, I don't know, or Nisha. Is there somebody doing these things here? I do note that one of these papers, this one, the first author is from Wildlife Institute of India. So that's at least one person who's working on it. But who else, I don't know.
No, but she has stopped completely working on that. You know, we contacted her.
I see.
She's
Okay.
working on something completely different now.
Okay.
Question?
Sorry, you showed a bunch of trees and you showed how they sort of spread out. But calculating distance from going one node to another node through the number of paths and distances of each, does that give any additional information?
In phylogenetics, it can give information. So we have been looking at selection pressures and non-uniform stationary states and so on. So for those calculations, we need the branch lengths. If you just want a tree, it still gives you the best tree because you're trying to calculate the most likely tree given the sequences that you do have. So the likelihood formula has those lengths.
No, what I meant was you have A, B, C, D, E as 5 elements, and then you have the tree which tells you how the spread happened. Thus calculating, say, A to T distance as, you know,
so you're saying, do we need these numbers Q here, or you just need a topology?
Yeah, Q1 plus Q4 plus Q3 kind of a thing for distance between A and T and so on.
Yeah, I don't know whether you need those numbers, but you need to calculate them to get the likelihood, is what I'm saying. You need them for certain purposes. If you're looking at certain evolutionary problems, you need those numbers.
I think there's a question there.
This phylogenetic tree and Nisha's hierarchical clustering, right? So do they use the same algorithm over here? Or
I think what Nisha did was probably something like UPGMA, which is called agglomerative clustering, but maybe, yeah. So it is a, in UPGMA you have, you use the pairwise average as the distance between clusters, but there are, in clustering generally, you can use the average distance, you can use the closest distance, you can use the furthest distance. There are various approaches to do it. I forget what those are called. Nisha can answer what she used, I guess. Yeah. Thanks. But it's the same idea, basically.
I mean, what you said is correct. I mean, so we, I think it was the average distance. I mean, if I remember correctly, but there are a couple of options. We can choose. You can actually run your experiment and see. I mean, the various options and what result you get. But the distance measure, yes, you have a choice over there. You can actually choose among the various distance measures.
Yeah. So in
it was agglometric clustering, so certainly. Yeah.
Right. You can use Euclidean distance or other.
Yeah. Euclidean distance also one. Yes. Will slightly change. Yeah.
It will.
It will slightly change. It will not remain. Yeah. But overall.
Yeah. So this is a sort of textbook example. UPGMA assumes what's called a molecular clock. Supposing that is not true, so your tree structure actually looks like this is your ground root structure, as in from the fossil record, you know, this evolved to this, but this for some reason found its niche and stopped evolving and thus kept evolving. This can happen. And then UPGMA will actually, if this is ABB, CD, then UPGMA will give you BC closes. And then it will give you maybe A and D. So this is a case where it can go wrong. So it depends on the assumptions. And this is a case where neighbor joining gets it right. So that's the usual motivation.
Any other questions or comments? Okay, if not, let's thank Rahul once more.
Thanks.
And now we break for lunch, and please be back here by 2 o'clock when we have the talk by Professor Mayank Vahia.